I requested a challenge response for Exabyte Spyder in regards to my Convergent evolution versus universal common descent video, and given the opportunity to falsify my arguments that were against the evolutionary assumption that common inheritable traits were a great indication for common ancestry. He accepted the challenge fortunately, and eventually made a video titled Melting Ice. In his video, he disputes my arguments by implying a variety of points. So let's get started with one of his major arguments. Each homology, if common ancestry was true, should be present to its corresponding phylogeny as opposed to others. So a falsification for common ancestry will be if homologous features were to be present across different evolutionary branches, because that would defy otherwise the unique patterns of similarity.
Actually, falsifying common ancestry would be more than just reframing homologous traits into analogous traits. To really falsify the idea of common ancestry as predicted by evolutionary theory, you'd have to positively falsify the facts that 1. Genetic changes occur with every generation, 2. These changes are inheritable and thus accumulate, and 3. The immediate environment places selective pressures on organisms to adapt or die. From these 3 facts that most would agree are true, without any other facts placing necessary constraints, we can derive inferences about what traits our ancestors must have had in order to give rise to their living descendants that we would see today. And these inferences are backed not only by comparative anatomy, but also by phylogenomics and the fossil record. So again, Ice's attempt to reframe homologous traits into analogous traits will not falsify the entire idea of the common descent.
Really, Exabyte Spyder. So, if one were to demonstrate homologous features coexisting across distant evolutionary unrelated species, then it will still not falsify common descent. Well, let's hear what the evolutionists themselves had to say about that.
When you look at a phylogenetic tree of life diagram, derived from the idea of common descent, you immediately notice the pattern of groups within groups. This nesting is unique to the common descent idea. The statistical analysis of these characteristics of organisms grouped with this idea is unusually successful. Species are essentially never found that combine characteristics of different groupings. For example, snakes and lizards are never found with feathers. Only birds have feathers. Birds and crocodiles are never found with differentiated teeth. Only non-marsupial mammals have a placenta. Birds and mollusks are never found with placenta. Only mammals have hair and mammary glands. Amniotic eggs are never found in fish or arthropods. A mix and match of characteristics like these would make it extremely difficult to objectively organize species into nested hierarchies. The consistency of nested hierarchies is another success for common descent.
Similar. And so what evidence did Darwin use to back it up? Well, he started with fossil evidence. And so if you look at these 4 types of animals, they all have the same bone structure. And so even though they look different, like a human arm, a dog leg, a bird wing, and a whale flipper, they probably had an ancestor that had similar bone structure. And you could also see it in the fossil record. In the rock itself, you could see fossils changing over time. So horses, for example, as they moved out onto the plains, got larger and larger and larger, and you could see the change in their fossils. A big piece of evidence we have today is molecular evidence, so DNA evidence. And if you look at the organisms on our planet that are most related to us, we can line up our DNA and we can compare it to their DNA. And if you line up human DNA and chimpanzee DNA, you'll find that there's only 1.2% difference in all of their DNA. If you look at a gorilla, that jumps to 1.6, and in a baboon, it jumps to 6.6%. And so we can compare how much our DNA is alike, and I mean exactly alike, and say, well, we can see who's related to whom.
So, as you can see, Mr. Exabyte Spyder, even the evolutionists tend to disagree with you that common descent can only be falsified by the 3 points you mentioned: Contrary to your damage control logic, contradicting homology will be a big blow to the common descent theory, since it will be quite difficult to distinguish homologous structures derived from common ancestry from those that are mimic and originated separately without sharing the same or common evolutionary origin. So, homologous-like structures from species that originate from different ancestry rather than common ancestry will be a devastating blow to the evolutionary theory of common descent, since they arose independently rather than by the common inheritance from one generation to the next. Even the website that which you listed seems to also disagree with you, as quote-unquote, if it could be shown that when mutations do occur, they are not passed down through the generations. 2. If it could be shown that organisms with identical DNA have different genetic traits. There are two things I can conclude here so far. Either Exabyte is not aware of these falsifications, or he is just being outright dishonest in order to save common descent.
The burden of evidence is still on him to show why all similar traits both in living and extinct species are analogous rather than homologous. He has to show why modern classification backed by phylogenomics and comparative anatomy is inherently flawed in making inferences about common ancestry. In other words, ICE has to show where the evolutionary tree of life fails, where some or many of the organisms that are agreed to be in one group are not and cannot be grouped with another group of organisms that share other characteristics in common that evidently show a common ancestral link. He must also show why ERV ratios within the genomes of related species conform exactly to what common ancestry would predict, in addition to the genetic similarity in general, not of just all animals in the animal kingdom, but also the similarities among the kingdoms of animals, fungi, and plants, which only points to an all-encompassing eukaryotic origin for all multicellular forms of life. If he denies that such an extrapolation can be made, he must show where their taxonomic classifications must fail. Where is the threshold that keeps one group of animals from not showing common ancestry with another group of animals?
Now, playing the devil's advocates here, Mr. Exabyte Spyder claimed that one way to falsify phylogeny classification in favor of common descent was to demonstrate phylogenetic trees that group species in the wrong place or give out contradictory results. He affirms this by relying on an affinity of morphological and genetic inheritable principles that can be used to establish evolutionary relationships and its history. Now, contrary to Exabyte Spyder expectations, there is actually already evidence where phylogenetic trees based on morphology tend to give out contradictory results compared to molecular analyses. Back before molecular phylogenies were introduced, classical taxonomists have created evolutionary models where species were grouped based on homologous structures, which, according to Spyder, must be evidence that those structures derive from common descent. According to Douglas in 29 Plus Evidences for Microevolution article, he claimed well-determined phylogenetic trees infer from the independent evidence of morphology and molecular sequences matched with an extreme high degree of statistical significance. Yet, in reality, the technical literature tells a different story. One of these significant cases was known as the Cilimata hypothesis. In the Cilimata hypothesis, expert morphologists of taxonomy have constructed evolutionary history based on the similarities in which they regard as homologous while distinguishing those from analogy. Taxonomists in favor of the Cilimata model have considered the presence of cilium body cavity that is present in both arthropods and vertebrates as homologous, indicating that they share a common ancestor that possess those homologous traits. Yet, around the mid-1990s, a very different arrangement of the species animal groups were proposed based on the analysis of molecular phylogeny. A team of researchers who proposed this arrangement published a groundbreaking paper in the peer-reviewed journal Nature with a title that surprised many taxonomists: Evidence for a Clade of Nematodes, Arthropods, and Other Moloeating Animals. The paper noted the conventional wisdom of the Cilimata model, mainly that both arthropods and vertebrates were closely related because both phyla had segmented body plans. However, an analysis of the 18S ribosomal RNA suggested a different grouping, one that placed arthropods closer to nematodes that interestingly do not possess homologous features, typically the cilium, as the arthropods do. The Nature paper demonstrated this unexpected grouping as quote-unquote, considering the greatly differing morphologies, embryological features, and life histories of the molting animals, it was initially surprising that the ribosomal RNA tree should group them together. Even Wikipedia has regarded the Cilimata hypothesis as a dead-end evolutionary model, despite the fact that it was based on homology with the expert separation of analogous structures. I encourage Mr. Exabyte Spyder to read the paper by Develos titled Understanding Phylogenetic Incongruence Lessons from Bats, where the paper quotes incongruence between phylogenies derived from morphological versus molecular analyses and between trees based on different subsets of molecular sequences has become pervasive as datasets have expanded rapidly in both characters and species for more information on why phylogenies based on homology often or sometimes tend to give out inconsistent evolutionary history compared to molecular ones. The fact there are morphological phylogenetic trees that do not absolutely agree with molecular ones says a lot against Exabyte Spyder's claim that homology is a great indication for common ancestry. Of course, there is no dispute over the fact that pure morphological classifications show very clear evidence of less complex species evolving into more and more complexity with new phyla if we compare our homologies in respect to ancestral divergence, as what would be expected if the tree of life actually existed. Yet, Exabyte Spyder's credibility of his homology argument in defense of common ancestry becomes rather his own upsetting dilemma over the fact that phylogenies based on nucleotide amino acid sequencing or DNA often tend to give inconsistent and contrary results compared to that of morphological homology. This fact demonstrates two remarkable things: that one, homologous structures can also too independently arise from different genes rather than inherited ones from the same common ancestor, and two, demonstrating that establishing true evolutionary origin has proven to be quite difficult due to phylogenetic incongruence. But I'm being generous here. Now, even if Exabyte Spyder argued that molecular trees show true ancestry because of genetics, that argument too doesn't work because constructed evolutionary trees based on a variety of genes also tend to be proven quite elusive. In Roca's 2005 paper, Animal Evolution and the Molecular Signature of Radiations Compressed in Time, the study has sought to determine the evolutionary history of the animal phyla by analyzing about 50 genes across 17 taxa. At the same time, he hoped that a single dominant phylogenetic tree would emerge. Rokas and his team reported that a 50-gene data matrix does not resolve relationships among most metazoan or multicellular species, because it generated numerous conflicted phylogenies and historical signals. Their conclusion was unambiguous. Despite the amount of data and breadth of taxa analyzed, relationships among most multicellular species remain unresolved. Instead of them establishing true evolutionary history, they constructed inconsistent molecular phylogenies that contradicted evolutionary history. What was the cause of such incongruent phylogeny? The answer to that was incomplete lineage sorting and rapid divergence. Scientists like Rokas realized just how difficult it was to get high-resolution relationships when the divergence between them was extremely short, like during the Cambrian explosion, as is being researched in the paper. Actually, the paper is based on the Cambrian explosion. Both Rokas and Kerbel try to explain the many contradictory molecular trees by proposing that the animal phyla might have evolved too quickly for the genes to record some signal of phylogenetic relationships into their respective genomes. In their view, if the evolutionary process responsible for anatomical novelty works quickly enough, there will not be sufficient time for differences to accumulate in key molecular markers, in particular those used to infer evolutionary relationships in different animal phyla. Therefore, when groups of organisms branch rapidly and then evolve separately for long periods of time, this can overwhelm the true historical signal, leading to the inability to determine true evolutionary history. So, unfortunately for Mr. Exabyte Spyder, his argument that using genetics to determine true evolutionary history doesn't work either, since the rapid divergence of species has corrupted the genes to establish a high-resolution evolutionary history phylogenetic tree. Another article titled Convergent Adaptive Radiations in Madagascan and Asian Renin Frogs. Researchers report both the Madagascan and Asian frogs to share identical ecomorphs, such as remarkable similarity in morphological, physiological, and developmental characteristics through independent parallel adaptive pathways. Despite such remarkable similarity, the researchers conducted analysis of nuclear and mitochondrial DNA showing that the two different species of frogs are actually unrelated to each other, sharing no common ancestry at all. Although the frogs were shown to have ecomorphs together, this isn't the case for all rennet or reined frogs.
The fatal flaw in Nice's interpretation is that just because the researchers indicate convergent evolution with these particular species of frogs as a result of similar natural pressures, it does not immediately indicate nor even imply that the two frogs are wholly unrelated. Not only did the researchers not indicate this, but if you look at one of the accompanying figures, you'll notice a cladogram of the two groups of frogs, connected by this line in the center. If you know how to read cladograms, you'll understand that this line represents a common ancestor between the groups, thus denoting common descent for the two. Why would the researchers show common descent, despite also demonstrating a case of convergent evolution? Probably because common descent and convergent evolution are not immediate opposites.
Starting around 8:40 in this video, Mr. Exabyte Spyder claimed that the Madagascan and Asian frogs are closely related based on the line of the cladogram as you have seen there. Sorry, Mr. Exabyte Spyder, but you are wrong. This is what the researchers actually said regarding the two frogs. Our results do not support the traditional grouping of ranids according to ecomorphs, but rather reveal that the relationships were primarily shaped by plate tectonics. Indeed, the inferred phylogeny exhibits a branch separating a Madagascan from an Indian group despite the fact that identical ecomorphs are found in both groups. MP Bootstrap's analyses of the mitochondrial DNA, nuclear DNA, and mitochondrial DNA 1 datasets all yielded support for the branch, which yielded a 98% support for the branch separating Madagascan from Indian ranids. Apparently, as the authors have suggested, molecular phylogenetic analysis between the Madagascan and Asian frogs does not support the traditional phylogeny inference of them being closely related. Rather, the phylogenetic tree has grouped them away separately from each other rather than as closely related cousins, despite the fact that they appear to possess the same homologous-like morphology. This is pretty obvious if you actually look at the cladogram from figure 1 in the paper. His claim that convergent evolution can also be distinguished and compatible from that of morphological homology as to avoid deception in which species share a common ancestor or are closely related is also false. Even the researchers from the same Madagascan Asian frogs paper disagree with you, as quote-unquote difficulties in detecting convergent ecomorphs without the use of molecular data are particularly common in Anurian amphibians because their body plan is remarkably conserved. So, without the utilization of molecular data, it will be quite difficult to distinguish convergent evolution from homology merely on the basis of morphology, like body plans and other inheritable-based principles. What this paper demonstrates is that those homologous-like structures that can be quite deceptive without the use of molecular data can actually arise from different ancestry rather than a common ancestor. So, the Madagascan and Indian frogs actually arose independently and separately from each other. If those species were actually closely related, then their DNA would be very similar, that the phylogenetic tree would group them together. But the fact that their DNA, especially their mitochondrial and nuclear DNA analyses, are quite remarkably different, then they are considered distant species and considered to be unrelated from each other, which indicates that they come from different origins rather than a common origin, which that common origin is known as the common ancestor.
One can make the same argument for why manatees are sometimes born with hooves, why humans and other apes have folds in our ears, and even why many males of placental mammal species have nipples. These are all vestigial structures that denote common descent as a result of homology.
This second argument presupposes, in defense of common descent, vestigial structures or organs. This argument proposes that vestigial features are apparent to be mostly useless, although may still be functional to some degree. It is to a great indication for common descent since it demonstrates that the structure used to be once functional. There are serious problems with this argument, though. First, the vestigial argument already presupposes a common ancestor rather than actually demonstrate it, or at least demonstrate its origin or its so-called original functionality from the same or common ancestor. Second, the claim that vestigial structures are mostly useless begs the question since it relies on the presumption that it is non-functional rather than the fact, since one cannot possibly tell with any assurance that an organ is actually vestigial or not. It may as well be that we have yet not found its use. A vestigial feature that now seems to have no apparent function may in the future be found to have a function. Contrary to evolutionary claims, this is exactly what the history and the science demonstrates. Functions have continually been found for organs that were actually once thought to be vestigial. About 120 years ago, Robert Wiedersheim published a list of 86 human organs and anatomical features that he called vestigial. Gradually and ironically, one by one, they were later found to be quite functional, exposing the logical fallacy of the vestigial structure or organ fallacy argument in favor of common descent. According to evolutionists like Exabyte Spyder, there are still vestigial features present, but regardless, the trend is pretty clear. The vestigial argument list is actually being gradually exposed and constantly shrinking even more tinier over time. I encourage him to watch my evolution veritor argument from Poor Stupid Design video to show just how fallacious the vestigial thinking really is.
Again, these are not analogous limbs because all of these limbs serve different functions for their respective organisms. These are structural similarities that transcend the functions that they offer, and as such are homologous, thus indicating common ancestry. Let's consider what a comparison of analogous traits actually entails. Take the previously mentioned examples, the wings of a dragonfly, the wings of a bird, and the modified fins of a flying fish. All of these creatures have adaptations for flight, but these adaptations vary massively in structure, appearing only as a product of similar natural pressures that the animals adapted to. The dragonfly has thin, transparent wings made of chitin, which differs markedly from the typical bird with wings lined with scales and overlapping feathers, supported by calcified bones in a tetrapodal arrangement. The flying fish's fins are structured like a rayfin fish, and their similarity to other rayfin fish are noticeable despite the anatomical modifications they needed to get the ability to take flight. This phenomenon of species adapting to similar functions using their inherently different structures is called convergent evolution, and all three of these traits that their respective animals use to attain flight can only be called analogous because despite the function being the same, they're structurally far removed, preventing them from being called homologous wings. Therefore, no common ancestor would exist that would have given these organisms their flying apparatuses. However, this does not immediately mean that all three of these creatures do not have a common ancestor at all, because there are other traits, structural traits, that they do have in common with each other, regardless of whether they have the same function or not. Some homologous traits that dragonflies, birds, and flying fish have amongst each other don't include their flying apparatuses, but rather include cell structure, multicellularity, and tropoblastic development. It's in these similarities that a common ancestor can be perceived, although one would be hard-pressed to find that one ancestor specifically, at least in the fossil record. Since a common ancestor can be perceived to have passed down these traits to the more immediate ancestors of the dragonfly, bird, and the flying fish, and their closer cousins, it's thus fair to say that it's those traits that are homologous, not their limbs. Because the terms homologous and analogous can only be applied to specific traits, not the entire animal species nor the immediate ancestors. One trait can be derived to achieve a certain purpose, thus denoting an analogy, while the same animal might have another trait or multitude of traits that can only be homologous. This is what Ice is forgetting, and in omitting such a crucial detail, he's guilty of cherry-picking. Apparently, it's simply inconceivable to him that a descendant can develop traits similar to its cousin both as a result of having adapted to similar environments and having been derived from a common template, i.e., a common ancestor. That's a dichotomy that shouldn't exist, because natural selection doesn't care what traits the organism already inherited from its ancestors. It only forces the population it acts upon to adapt or die. And if two distant lineages end up being in similar survival situations, it really shouldn't be a surprise that similar adaptations would appear as a result. Like how the shark, ichthyosaur, and the dolphin employ the same general shape for swimming in the water, despite being on very distinct branches of the evolutionary tree of life. Their tail fins are another example of analogous traits. They achieve the same purpose, but are structurally far removed from each other because of their immediate descent from different ancestors. However, they have other traits that imply their immediate ancestors to be related by a common ancestor themselves. So if sharks, ichthyosaurs, and dolphins are related to their immediate respective ancestors, and their ancestors are related to each other by an earlier common ancestor, then sharks, ichthyosaurs, and dolphins are also related by the same common ancestor, and this ancestor exists regardless of what we would call such an ancestor. Ice's biggest point he's trying to bring across with this video is the idea that since analogous traits can arise, it is thus just as valid to call the traits exhibited by extinct forms analogous rather than homologous to the traits of their descendants. There are some problems with this thought, the main one being that in order to show that living organisms can indeed develop massive similarities without coming from a common ancestor, you must show exactly what the mechanism is that allows such a thing to appear in such great magnitudes. So far, you've only been able to show that convergent evolution can occur for a few traits within a family of organisms, not that convergent evolution can create a whole new organism with all possible traits not derived from a common ancestor with other organisms. You must show that it's not only possible that separate lines of evolutionary change can turn up so many genetic and morphological similarities among unrelated creatures. This includes being able to breathe oxygen instead of any other gas, having eukaryotic cells with the same organelles, having the same structures in the same form regardless of function, etc., but also that such a scenario is more likely than traits simply being inherited from common ancestry.
This is going back to, in regards, homology and analogy. I apologize to Exabyte Spyder if I wasn't clear enough on what I meant by morphological principles, which was to say, in other words, inheritable morphology that were presumably passed on from one generation to the next. Now, regardless of his distinction, I believe I don't have to further elaborate myself about this since I already demonstrated earlier how homologous features can also arise from independent and parallel lineages that have nothing to do with the inheritance of common ancestry. One thing to remember, Mr. Exabyte Spyder, is that just because two or more species appear to possess segmented body plans doesn't necessarily indicate that they share a common evolutionary origin. It could quite be that they originated from their own different separate lineages as opposed to the common ancestor derived from the common lineage. That is presumed by evolutionists. The fact molecular genetic DNA analyses tend to give out contradictory evolutionary relationships compared to that of segmented homologous body plans really says a lot against the argument of using homology to establish ancestry. Even among the lines of genetics, it remains quite elusive to establish high-resolution phylogenetic trees due to the fact that the rapid divergence from one species to the other has corrupted the genetic key markers and eventually rendered it useless as a means to establish evolutionary phylogenetic trees. This says a lot against the so-called gradualism theory of evolution, since molecular analyses clearly demonstrate that multicellular species first originated very rapidly during the Ediacaran explosion, otherwise known as the Avalon explosion, which predated the Cambrian explosion, which was also a rapid divergence event where more complex multicellular species also arose. Thanks to the rapid divergence events from the explosive events, establishing common ancestry based on sheer molecular phylogeny has rather become an evolution fairy tale rather than a reality. Well, that's the end of my presentation in response to Exabyte Spyder, so thanks for watching.